Hi everybody, hope you're doing okay in the current circumstances and keeping as safe as possible. I think that's the main thing, is to look after yourselves and all your family and friends as best you can. I hope I suppose your FaceTime or your different apps for connecting with family have got well used in recent days. So I wanted to catch up with you, I suppose, in the module. I sent you a video last week just to give you an overview of kind of the lab, the lectures and the CA and things that were outstanding and kind of where we were. I know this week is a study week, so what I wanted to do was put together some resources for you, maybe on some of the topics we've covered already as a revision. And then in the coming weeks, I'll be able to share with you information on the topics that we have left to cover, those that I mentioned in the email. So here, I thought we could start with something that we've covered in a lot of depth called replication, DNA replication. And to do that, I thought that I'd be able to maybe look through some of the key slides and remind you of some of the key points. So if I was you, what I would do here is get the past papers that you can access online and see if there's any questions on, say, replication that you can review and see how you feel you could address them. And maybe a good idea would be to stop the video after I've introduced it here and maybe look up those papers and review them even mentally and see how you feel with that topic. And then obviously watch the rest of this video and the different resources I'll send you, and then maybe revisit that question and see how you feel about it then. As I said, the main priority is you and your friends and family. And I know it can be a very anxious time moving to different kind of delivery methods, and even just the idea of a change in routine, that attending classes, attending labs, seeing your friends, and now working from home and potentially in a form of isolation as well. So if there's anything you think that I can help you with or support you with as a group, feel free to let me know or contact me. I'll do my best over the coming weeks to support you as in every way I can with things that I feel will help you go back over topics and help you understand future ones. Okay, so let's lead in with our overview of replication. Look over some of the key slides and let's have a little review of, say, the process and how the replication fork is built. Okay, here we go still.
Okay, so here's our molecular biology DNA replication handout that we went through. And even by the image on the front, you can see the dark blue strands are the parental strands and they're being separated permanently. And then there is these purple strands being made using the navy strands, or dark blue strands, as a template. And that's effectively our semi-conservative process. So I thought it might be good to go through this handout, just a few of the key slides that are in it, just to kind of refresh your memory and revise the topic. So you would have looked at these as part of the handout and in the classes we went through. So even as we go look at the scan through the slides that are here, you'll see there's a few key images that we can refer to help us with our understanding. So for example, we went back to the cell cycle and we reminded ourselves of these G1, S and G2 phases that occur before mitosis. So every time a cell goes through mitosis, which you know well from previous modules, the cell divides into two, and then those cells are going to go through G1, S and G2 phases before going through M and so on, dividing into two cells, and the pattern continues. So this is known as the cell cycle, and we spent a lot of time talking about this, discussing the idea of checkpoints, discussing the idea of making sure damaged cells don't go through mitosis and so on. But this is the key phase that we're looking at down here, DNA replication in S phase. So S stands often, if you remember, for synthesis, because we're synthesizing new pieces of DNA. So the S phase is where we're going to duplicate the DNA or the genome. So essentially photocopy what we already have so that we can have two copies identical in every way that we can pass on to the two daughter cells later on after mitosis. So it's that copying mechanism. So can you imagine at the start of S phase, you'd be copying, say, a third. In the first part, about a third of the DNA is copied. Here we'd have about 2/3 copied, and then finally towards the end, you'd have it all copied. And once the DNA is copied, we're able to progress into the next phase. And we talked a little bit before about making sure how cells don't jump into the next phase prematurely and how we regulate the cell cycle and the transition from one to the other. So where is it going to take place in the cell? Well, it has to take place in the nucleus, because in the nucleus is where the DNA is housed. So all the machinery that we need has to be available within the nucleus for replication. So if we consider, I suppose, all those different processes that we've talked about so far, you'll see replication here at the top, okay, in this diagram, where our DNA in one long kind of strand is being duplicated into another copy. So even in this diagram, you can see the idea of the parental strands being separated on each side, and you see these red strands being made, which represents the new daughter strands in this diagram. And then these DNTPs we referred to before, the kind of building blocks, the G's, A's, T's and C's that are going to be used. So replication, everything gets replicated. It's very different than, say, transcription, where we turn on certain genes and we don't turn on other genes and so on. This is where we need to copy absolutely everything, okay, the whole library that we have in the genome. So if we go back and say, well, what other key images can we find, or what other key points did we discuss? So I'm just trying to bring you back to these slides so that you have that familiarity when you're going through again. This slide, we discussed the idea of how replication occurs. Did it happen by a conservative mechanism or semi-conservative? So we've discussed that a little bit more even today here. The idea of semi-conservative, where the parental strands, the red ones up in the top right, are permanently separated, and we use them as templates to make the new daughter strands. And they in turn will be separated and new strands made off those as templates, etc., etc., etc. So semi-conservative was what I asked you to fill in this box so you remember that aspect. So what have we done so far? Well, it happens in a phase of the cell cycle called S phase. It occurs in the nucleus, and it's a semi-conservative process. Okay, so replication, we've learned a little bit about those already. So how does it happen? Well, the first thing I brought you through was this idea of a particular enzyme, and that enzyme was called DNA polymerase. Now, we're talking very generally here. We learned a few phrases later on that they were called different things in bacteria or in eukaryotes. But if we knew the sequence of one strand, we could actually realize the strand of the other. And this was something that Watson and Crick presented as well, this idea that there must be a copying mechanism based on a template. So using the base pair rule, if we know one strand is GC, 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 well, we know the other one is CG, 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 and so on. So we've talked a lot about that in class. But these enzymes, these DNA polymerase, they have certain features that we said were very important. So we said that they shape almost like the shape of a palm. And in that kind of region of a protein, there are different zones or different regions for different functions. So we have part of that protein where we're going to be able to extend and read the template and put the right base in the right place. So up in the top left of this slide, you see this blue strand, this parental strand has been separated, and we're using it as a template to extend the light blue strand. So if that's a G, we'd put a C. If that's an A, we put a T. T, we put an A, and so on. And then at the bottom, there's something called an exonuclease site. And this is to do with its proofreading ability. So one of the key features about this enzyme is that it has proofreading ability, and it's a very template-driven enzyme. So when it sees a mistake, then if it makes a mistake, so it always kind of, the analogy people say was it looks over its shoulder after it's put a base in to make sure they've made the right connection, G to C or A to T. And if incorrect, they can flip the strand. So if you look at the diagram on the right, they flip the light blue strand down towards the bottom, towards this exo site, right? This cutting site effectively, where they can cut off the mistake, and then it can flip back up to where it used to be, like in the left side diagram, and continue error-free. So I hope we've discussed quite a lot the importance of making sure that we don't have a mistake. In the first handout, we talked about the issue with mutations and all different types of mutations that can happen and what occurs if that's the case. So if we come back again, we say this is a super enzyme, an absolutely amazing enzyme, template-driven, proofreading ability, but it had a massive disadvantage. And that's kind of what I want us to kind of consider a little bit now. Okay, so the problem with this enzyme, effectively, is that it can't start from scratch. So unfortunately, it's not one that can start building a DNA chain from one base to another, to another, to another. It requires a template, firstly, but then it also requires something called a primer. And a primer is a very small, single-stranded sequence of nucleotides that it can extend. So if we can lay down a primer, then we're going to be able to extend that primer. And that's what DNA polymerase can do really, really well. But it can't start from scratch, or it can't initiate the process. So what we discussed before was borrowing another enzyme which has that power. So the cell uses RNA polymerase, which is able to initiate RNA synthesis without a primer. And we talked a lot about, well, if this is RNA, what are we going to do in the process? So that's something we might just review as we go through the process itself. But remembering now, reminder that DNA polymerase does have a negative or a disadvantage, but the cell overcomes that problem with a solution. So effectively, we're going to get, in an overview of process, we're going to unwind our strands. But as we do that, sometimes we can get a lot of tension building up in the particular strands itself. So we watched a video about these knots and twists and the role of topoisomerase enzymes and how they can relieve torsional stress in the DNA built up from separating the strands. We talked a little bit about helicases. They're hexamer, so they have 6 subunits and they use energy to pull the strands apart. They use ATP to pull apart the strands. But we wanted to focus initially on how did they know where to start working. And we call these regions origins of replication. And those origins are very AT-rich sequences. So they're like landing sites, effectively, for helicases that pull apart the strands. And once we have the strands pulled apart, we can start holding them apart by using certain proteins that will stop them from re-annealing back together, the strands reconnecting. And then we're going to start this primer process that we discussed. We'll come across a problem due to DNA's anti-parallel structure of leading and lagging strands and these Okazaki fragments we have to join together at the end. So I showed you a couple of diagrams here with the DNA replication fork, and I think it's important to spend a bit of time actually really studying this one and the next one, because it's often where students find the concept a bit difficult. So it's all to do with where you put your 5s and your 3s. And if you think through the process, instead of trying to learn off a diagram, instead of trying to memorize scan it into your brain, into a folder, I think you're much better actually understanding the process like we've gone through. So I showed you this diagram first, and we said, look, there's a strand, there's a leading and a lagging strand, and leading was a continuous strand, and lagging was made up of these little bits and pieces that were going to be, I suppose, fused together later on or joined together. But we call that discontinuous, made up of these Okazaki fragments. So then we looked at this, which is a bit of a, I suppose, a simpler diagram. But I just want you, because there's no coils and so on. I just want you to follow the 5s and the 3s, okay? And that's the key thing. Wherever you put your 5 on one strand, 5 to 3, the other parental strand must be 5 up at the top, down to 3. So remember, DNA is anti-parallel. Each strand is going in a different kind of direction. So 5 to 3 and 5 to 3. We spent a bit of time on that. So if we separate these strands like we're seeing in the fork, and we want to make new strands off those as a template, well, they can only be made in a 5 to 3' direction. The enzymes only work on the new, they synthesize in a 5 to 3' direction, extending the 3' end of the primer to make the bit longer, the primer longer. So if you watch the red blocks are the primers. So let's take the bottom strand. We have 5' over here in the light blue, down to its 3'. So that primer, we now know the 5' end and the 3' end of the primer. And the 3' end of the primer is now going to get extended and extended and extended. And it's moving in the same direction as the fork is opening, I suppose, by the helicases. So that way it can be made in a leading or continuous fashion. The other strand's a bit different. If we look at the 5 to 3s on the parental strand, 5 is up at the top, down to the 3. And because of that, our primers, once they're laid, we know this is the 5' end here and the 3' end here. So we're synthesizing new DNA in the opposite direction to which the fork is moving. So we make them in small little fragments that almost bang into each other, essentially, won't they, as they get extended. And as we unravel or open up the rest of the DNA helix, we'll be able to just, on the leading strand, keep going. But in the daughter strand, we have to lay down another primer and extend it in the opposite direction that the fork is moving. And then finally, we'll join all these fragments up later on. But before we can do that, we have to get rid of these little red blocks. So I hope you can remember why. You do, yeah? Of course you do. They're made of RNA, because we used RNA polymerase or primase to lay down those primers. So when we went through, I suppose, the concept of the fork, and we realized there were a few problems, I gave you this table. And I really like this table because I think this will really help you appreciate your level of understanding. So what I would say to you here is revisit this. Look on the left-hand side and see, can you write a descriptive answer on the right-hand side for each one? Okay, and I think if you can do that, you'll realize the problems that replication faces and how the cell has overcome those to make sure it's a very functional process. Okay, so I'd like you to kind of revisit this particular one here, as well as understanding that replication fork. As we went down through the slides, we also looked at, say, eukaryotic replication in detail. And we started saying, instead of in bacteria, they call them polymerase 1 or polymerase 3. Here there was pol alpha and pol delta. And we kind of put names on the different proteins. Instead of single-stranded binding proteins, we started putting the idea of RPA in the middle, the replication protein A. Instead of the sliding clamp, which holds the polymerase on the strand and helps processivity, we called it PCNA. So these were all the kind of correct terminology, but I still wanted to show you that aspect. We learned about these replication processes by studying the SV40 virus and how it infected certain cells and how it replicated itself. So we kind of used a model system to learn a lot about how replication works. So I asked you about that table there towards the end, and you'll see that I kind of have the answers for you at the very end, but maybe give it a go first. So this was a slide that we did on a whiteboard down in NC106 a couple of years ago, and I asked the students to kind of give me all the key aspects of replication that they felt were important. So it's another nice slide that you can revisit in the notes, just to see is there some things you could add to it, or is there anything, the bullet points that you want to understand or read a bit more around. So with replication, I suppose there was only one other thing what we kind of mentioned towards the end. That was the idea of telomeres. And we talked about where they're located in the cell at the end of the chromosomes, and how we have an end replication problem, and how the very ends of the chromosomes, it can't be replicated. And every time we go through S phase, the ends of the chromosomes shorten a little bit. So we discussed that in a lot of depth. So I hope you have some ideas about how telomeres shorten like a kind of a clock almost as to the age of the cell, if you know what I mean, but the age. So every time the cell divides, it shortens the telomeres. And we remember what we discussed about if we reach critical lengths, what occurs within the cell. So I hope that gives you kind of, I suppose, just an overview of some of the key things that are in our slides here and key things to do with replication. Yes, you can fill it in a bit more with some of the details like the SSB or like the RNase H and so on, but I hope that gives you just a snapshot of how it works.
Okay, so if I suppose think of replication, there's one concept I want you to get used to or be aware of first, and that's the idea that DNA, if we take two strands, DNA is an anti-parallel structure. So that means that we will have a 5' to 3' strand connected via hydrogen bonds and so on, like we've covered before, and going from the other strand 5' to 3'. So it's really that idea of an anti-parallel structure, and that's something I really want us to grasp. Okay, is this just that we can clearly see that principle, because if we get that principle, then it's going to really help us understand replication a bit more. Okay, so let's go back to replication again with that knowledge and that awareness. So if we take DNA replication as the fork structure, and if we see these parental strands connected together, so we know already that these are connected via hydrogen bonds, but because they're separated by a helicase, these strands are now showing their base. I suppose we can read the strands that are here. But first, I want to do what we should really consider is 5 to 3. And if that's 5 to 3, the other strand is 5 to 3. Okay? So in this structure, if we had to draw where the helicase is, it's going to be located in this structure. And the reason I'm putting it here is I want you to see what would happen next. Well, it's going to start separating the strands and move in a particular direction. So which direction do you think the helicase is going to move in, left or right? Well, what we've looked at in the diagram before was the helicase will move left. So what we can do here is draw an arrow, keeps us remembering that, and we can say, let's make this a bit smaller, and we can write the direction of fork movement. Okay, so that helps us remember that that's the direction, and that direction is, I suppose, helps us later on when we start thinking about building new strands. So let's think about this bottom strand, for example, the one with the green numbers associated. Now that's 5 to 3, and in that structure we are going to have to lay down a primer to build new strands. And in the blue strand, we're going to have to lay down primers as well to make a new strand, but we want to know which way we're going to go. So I guess the rule to remember is the new strand has to be made from 5' to 3'. Okay, so let's start, I suppose, the easy one, the blue one at the top. If we say that this is 5' to 3', right? So we know on this strand that this is the 5' end, moving towards the 3' end. That means any new strand of DNA that's going to be built on this template has to be made from 5' to 3'. So if this is 5' to 3, any anti-parallel strand will have to be 5' to 3 in that direction. Okay, going from, as I'm indicating here. So let's draw that primer at the moment. We'll say in red. Okay, so let's put the primer here. Now we know with that primer, we know that this is the 5' end and this is the 3' end of that primer. And we know that what we're going to do is extend the 3' end. So if I draw, say in green, the new strand, we're going to draw like this. So this is the continuous strand. Okay, now you'll see text as well calling it the leading strand in textbooks and in diagrams. So why is it called leading or continuous? Well, it's moving in the same direction, right? It's moving in the same direction that the fork movement is moving in. So I want you to look at that green arrow here and that yellow arrow here. They're both kind of moving in the same direction. So as we continue to break the strands and move in, separate the parent strands, we can just continue building this particular leading or continuous strand. So now let's flip our attention to the other side. So on the green strand, we know this is the 5' end. So anything we lay down will obviously be moving in this direction, 5 to 3, right? That direction, 5 to 3. So let's lay down a primer, for example, here. Okay, so we'll say that's the primer. We know now that this is the 5' end and the 3' end of the particular primer. And if we extend the primer, we're going to make it in this direction. So we're extending the 3' end, but it's moving in the opposite direction than the fork movement. Okay, so I want you to compare this small green arrow here with the direction of the fork movement. So as we separate the strands a bit more, we're going to lay down another primer, and that primer then will have to be extended and so on. And every time we open up, we have to lay down another primer, and we're always moving in the opposite direction. So those particular fragments that are being made are known as Okazaki fragments, and that's made after the scientist Rei Okazaki that discovered this process. So that should show you a little bit about how to label a replication fork. What are the key things to remind yourselves of? Well, where you put the 5s and the 3 is vital. So I want you to get, I suppose, a blank page and start drawing out, say, what I have here in black lines and label them and see, can you draw the strands in the right place? So the key thing, I think, when I went around your class to look at all your diagrams, in my head, I literally am grabbing this 5 and I know that's 5 to 3, so the new one must be 5 to 3. And that's how I tell if that's 5 to 3, that's 5 to 3. That's how I'm checking your work. If I have a 5 here, I know that's 5 to 3. So I know this must be 5 to 3, 5 to 3, 5 to 3. And I know I'm making it in the smaller, discontinuous Okazaki fragments because I'm going against the direction of the fork movement. So I hope that video gives you a little overview of something that can help you with the process and how it works step by step. I mean, when you look back at the diagrams and the process after this, you've learned already that we have other factors involved, like the PCNA or the sliding clamp, that help move the DNA polymerase along at a faster speed, the pol alpha changing to pol delta, which is higher processivity, more speed, more accuracy. We know that the red here are the primers that are laid by primase, which are made of RNA or RNTPs, the GAUs and Cs, and we know that RNase H, or in eukaryotes FEN1, has to take out those red entities so that they can be filled in with green. So it's worthwhile kind of considering the process here and what the replication fork diagram looks like, but then also adding on the other steps that happen to ultimately yield our photocopy or our replicated DNA double helix. Okay, so we cover quite a lot in these revision sessions, but hope that gives you kind of a nice summary, and now when you revisit any exam questions, you have a good way of kind of realizing how you can answer them already with the knowledge that you have, and maybe you've been able to generate some notes from this now that'll really help you coming up to your exams and even just understanding the process. Okay, talk to you soon.